Hi everyone. My name is LeClear. I'm here from the University of Texas at Austin, where I am working on my PhD with Beryl Simpson and Craig Linder. I'm working on the phylogenomics of the Physiognomous using RADseq to resolve relationships and identity of introgression, identify introgression within Jatropha. With the subtitle, What I'm Doing in My Friend's Attic on the Oregon Coast this summer. So, my objectives here are to resolve the phylogeny of Jatropha using RADseq data. Probably all familiar with RADseq, a lot of people are talking about it, but it's sequencing sites adjacent to, or stretches of DNA adjacent to restriction sites. And you can generate matrices of thousands of nuclear loci. I'm going to use that data set and that phylogeny to explore topological incongruence among data sets and to compare different phylogenetic methods. So specifically, I'm going to be comparing maximum likelihood and coalescent-based phylogenetic inference and do some integration analyses on that. So most people aren't familiar with the genus Jatropha, unless you're a specialist in the family Euphorbiaceae, which is a well-known family insofar as it contains some important species such as the castor bean, ricinus, commercial rubber, Hevia. There's a whole clade of succulents that are similar to cacti in Africa, that's genus Euphorbia, and cassava, which is manihot, closely related to Jatropha. Jatropha is a genus of about 180 species. They're trees, shrubs, and herbaceous geophytes. And they're adapted to arid and seasonally arid environments. The other reason you might have heard of Jatropha is that Jatropha curcus, or Physiognomous, is an important species for biodiesel production. My master's work was on the ethnobotany and seed oil chemistry of this species. So my first love is kind of with ethnobotany, and I appreciate the call out to Richard Schultes. Definitely was an inspiration to become a botanist for me in the first place. So here's a very rough map of its distribution based on GBIF data, and what you can see mostly is two takeaways. Number one, that there are areas of diversification in Mesoamerica and certain areas of South America, seasonally dry environments, and that there's very little data on localities in Africa. There's actually a lot of collections. South Africa is a huge area, the Eastern Horn, maybe 30 species occur in Somalia and Ethiopia. And then it goes into Saudi Arabia and India. So, a rough map, but I did most of my work in Mesoamerica in the last 3 years. So a little bit about the taxonomy. Currently there are 2 recognized subgenera. Most of the species belong to subgenus Jatropha. The subgenus Curcus over here has about 45 species. The main distinguishing feature between these two is the degree of fusion in the corolla. This is kind of based on an old taxonomic treatment, but that's one of the hypotheses that I'm working to test with my phylogeny reconstruction. Also, this is a almost entirely Mesoamerican clade, whereas this clade occurs from southern United States into South America, Africa, and India. So, for my study, I collected samples from the field and herbaria over the years of 2013 to 2015. Managed to cobble together about 70 accessions that I used in my final analysis, some more which didn't actually have enough reads to make it into the final matrix, but 25 from subgenus Jatropha and 37 from Curcus, so kind of flipped from where the vast majority of the diversity is in these 2 subgenera. It's really, that's mostly because I just didn't have access to African material. So I had some biological replicates, some multiple individuals of some species, which I ended up dropping from the final analysis after determining that they're monophyletic. I included Johannessia, which was determined to be sister to Jatropha in the previous analysis. And then a small number of Croton and Manihot. These are in the same subfamily. If you guys remember last year, there was a paper from Eaton, Donna to in Spriggs, talking about how increasing the representation in clades can kind of help to balance your phylogeny, and that should increase the locus recovery in your final assemblies. It was an attempt to accomplish that. I think either there just wasn't enough representation in these other groups, or they're just too phylogenetically distant. One of the issues of RADseq with phylogeny reconstructions, how deep can you go phylogenetically before you're simply just losing your restriction sites due to dropout mutations? So these were excluded from the final analysis, and I'll show you why in a little bit. So just to validate that Jonessia was sister, because there was only one species of Jatropha and Jonessia in that original study, I did some Sanger sequencing. And then for my RADseq library preparation, I used double digest RADseq. This is based on the protocol of Peterson et al. I used EcoR1 and SPH1, and sequenced across 2 lanes of Illumina HiSeq. This uses a combination of enzymes and size selection to give you your library of fragments that you will then sequence. So to select an enzyme, did some digest, different combinations of my available restriction enzymes, and then what you do is you end up picking, this is a bioanalyzer, so the distribution of fragments in different sizes, based on size. And you want to pick something roughly in this area where you have few enough fragments that you stand a chance of sequencing them all. And this is basically based on, there's a published genome for Jatropha curcus. Using that, I roughly estimated how many reads I was going to need. And for my previous slide, I got about 1.5 million reads was my target from sequencing. So that should have given me ample coverage for my window, which is right in here. It's about 410 was my actual target. So my workflow is demultiplexing the reads in DML, and then the assembly, I used IPyrad, and I created 12 data sets based on 3 clustering thresholds for the similarity to determine something's orthologous, and then 4 minimum taxa settings. So that locus has to be present in 4, 8, 16, or 32 different taxa for it to make a final matrix. So 12 data sets. Phylogeny estimation using RADseq, 10 independent runs with 100 bootstraps, and then Tetrad, which is part of the IPyrad package, which does, it implements the SVD quartets algorithm for a quartets-based phylogeny reconstruction. And then I did some introgression, and I'm going to call it incongruence analysis, which is basically my way of saying exploring the topological incongruence among my data sets and my methods. The ABABABA test as implemented in IPyrad again. So I didn't want to go helter-skelter and just compare all possible taxa combinations to see if there was introgression. So I kind of formulated a logical flow to try to test whether there was introgression between things that I suspected was possibly introgressing. So if the position of a taxon is unstable in the phylogeny, that's a clue. If the 2 species that I think are introgressing are sympatric, that helps. And if there's any other evidence, morphological characters, there has been some previous work in the 80s trying to collect systematic information, doing crosses, controlled crosses between a lot of species. So a lot of these are able to cross and has been demonstrated as being able to cross. So then I sequentially removed, I subset my taxa to a clade that I was particularly interested in. And then I sequentially dropped taxa and reran the RADseq trees. And then also the Shen et al. Paper that Joe talked about earlier, I calculated, you might have read it, I calculated the change in log likelihood score for a gene, comparing 2 different phylogenies for a particular data set. Saw which one of those were the most influential genes for the 2 topologies. And I haven't gotten to this yet, so I don't have actually results for that step. But what I want to do is check to see if those particular loci, which might be associated with introgression and influencing a topology change, are absent in sister taxa in the clade. So that might be evidence like this really important gene jumped into this taxon and is pulling it together to another one, but it's not in any other members of that clade. Anyway, just need to consider the number of reads and recovered loci, basically missing data. So the gold standard for introgression, the Abba test calculates Patterson's D. The Eaton-Reed paper on pedicularis, it's a modification of this. Instead of counting the patterns, it's a probability of the patterns, but it's very similar. All right, so my results. Sanger sequencing shows that Jatropha, or Jonessia, is sister to Jatropha. This is RBCL, this is a chloroplast gene. ITS and EMB, these are both nuclear genes. This is the ribosomal spacers, and this is low copy nuclear marker. So Jonessia is recovered in both cases as being sister here, not sister here, but the important thing is it's not in Jatropha, which was one thing I was trying to establish. All right, so the number of reads that are recovered, highly variable. So across all my taxa, there's the mean is 1.3 million reads per taxon. The standard deviation of 960,000. So some had a lot of coverage and some had very little. Now, that did not necessarily equate to the number of recovered loci. Right? So this has been explored by some authors to see if there's a relationship, a phylogenetic signal to loss or to loci being recovered, as opposed to your ability to recover loci is simply a function of the number of reads that you got for that sample. And I found actually this is a really rough way of doing this. I want to do a phylogenetic GLS on this, but I was just kind of roughly showing these correspond to my outgroup taxa. So even though I got lots of reads for it, I had very few recovered loci. Probably a result of them being phylogenetically distant and simply losing those restriction sites. So these are three graphs kind of showing the amount of recovered loci. It decreases across the increasing number of minimum taxa. The parsimony and formative sites also decreases, and so does the missing data. So it ranges from about 30% up to 80%. So I got a good tree. I'm really happy about this tree, and I'm going to use it for a lot of stuff. This is the RADseq tree based on just a single data set, and all the clades that are not shown with the number have a bootstrap value greater than 95. So this is also the distribution of the average bootstrap value for each topology based on those different data sets. So very stable pattern here, and a very erratic pattern here. So comparing that to tetrad, the major clades were recovered. Sorry, that's a sequence that's up there. So a South American clade, an African clade, Caribbean clade, and subgenus Turcus. So this is all subgenus Jatropha here. And these are southern United States species that are biogeographically very interesting to me. So comparing the topologies from the two, the red isn't showing up very well, but you can see, well, no, you really just can't. But there's some red there. This is using the Distory package in R that shows topological disagreement. And so I subset these taxa, or these clades here, to do my further analysis. I identified one particular taxon, Conzatii, which jumps out as sister to the rest of the clade, as opposed to within this clade here. Here's the result of my delta GLS that I described earlier from the Shen et al. Paper. So you can see there's a wide distribution. It's kind of cut up here. This goes up to actually 30. So I removed the highest locus and these other three, and what you see is between here, Conzatii drops out. So somewhere in those four loci, there's a strong signal pulling it into this clade. Again, I removed these taxa. I found that when Sympedala was removed, Conzatii jumps in. So I did my Abba test. It actually did not show that Sympedala was the one introgressing, but Deganii. So there's my significance, my z-score that shows that this is the introgression that's happening. So my conclusions, sorry to rush through those really quick there. Subgenus is monophyletic, Jatropha is not. Backbone is congruent between my two methods. Removing, and then this process that I'm not quite done getting my clear story from is a good way of sort of trying to figure out what taxa are involved and what genes might be involved in introgression when we see topological distributions. The species that was identified as being introgressing is not actually sympatric with the other species, but it's actually probably extinct in the wild now, so who knows.